Thanks for the introduction. Thanks for everyone for signing on to hear about what we do at Proof. So today I'm the opening act for Dr. Amrit Singh, and we'll be talking about how we perform data integration at Proof. So our objectives for today will be to show you how to gain deeper insights into biological questions with data integration, how exactly we integrate data from various sources, and how to visualize your data integration for your collaborators and high impact publications. And speaking of high impact publications, I just want to start off by highlighting some of the work that the Proof Center has done over the last 10 years. And this just kind of shows that we can work in any disease state as long as you have the data. So we've done work in transplantation, asthma, neonatal immunology, and through all of these, we've gained a lot of experience in data integration. And so we've also developed an interactive tool integrative tool called Diablo, which I should point out is not the video game. But I want you to keep that in the back of your mind when Amrit starts talking about it later. So this slide just illustrates what type of data integration we can do. And some of this might be confusing, but I find that it helps to have an example in your head before we get into the technical details. So today we'll be talking about two concepts. The first one is P integration. So when we talk about P integration, we want to integrate data sets based on the predictors. So say your study has measured certain proteins from asthma patients and you use an ELISA, but you have a collaborator who also measured proteins from asthma patients, except they used mass spectrometry instead of ELISA. So in this case, what we might do is identify the common proteins measured on the ELISA and the mass spec, perform normalization and corrections, and then analyze that data. So this allows us to go from only 10 subjects to an additional 25 subjects. We can also do something called N integration, where we integrate data sets from the same subjects. So maybe you have a cohort of asthma patients and you've measured DNA, RNA, or protein from all of them. And the goal in N integration is to analyze these data sets together and identify the relationships between molecules. So we can identify relationships between maybe RNA and DNA and proteins or metabolites, where we might not have if we were just analyzing them individually. And obviously, these can include integration of clinical and demographic variables as well. So with these examples in mind, I'll get into some more technical definitions. So a lot of the data integration that we perform uses what most people call big data. So some of the commonly recognized types of big data in science are transcriptomics, proteomics, genomics, metabolomics. And one of the important terms we use in big data is NXP, or N by P. And this describes how the data is structured. So I'm going to repeat this a lot in the next few minutes in the hopes that you can kind of understand it. N is traditionally known as the number, which refers to the samples or observations or patients in the data. And then the P stands for predictors. And so this can be any type of data obtained from patients, gene expression, demographics, proteins, metabolites. So if you can imagine, we can integrate numerous data sets, whether it's integrating data from a lot of subjects and thus increasing our sample size, or integrating different types of data, such as genes and proteins, and then identifying relationships between them. And obviously with increased sample size or predictors, we can identify novel biomarkers and therapeutic targets. So this N and P thing can be confusing, even for us so-called experts. And I hope this diagram can help you understand it a little bit better. So we typically have this NXP matrix where N is the samples and P is the variables or predictors. And so on the left, you'll see that we have N integration, where integrating data taken from the same subjects. So N is the constant, but the P variables differ. And in that asthma example I started off with, this is like if we integrated the DNA, RNA, and protein data together. In the literature, you might see this referred to as multimodal or multi-omics analyses. And then on the other side, we have P integration, where we find the same P variables between groups of different patients and integrate them to essentially increase the sample size. And so in the earlier example, that's like if we integrated your plasma data with a collaborator's data. So it's the same variables, but measured on different samples. And in some ways, this can be called a meta-analysis. So all of the N and P integration tools that we use have been combined into a single R package that Proof has developed with collaborators in Australia called mixOmics, with N integration being under Diablo and P integration being under Mint. And you can see that it's been cited quite a few times. And my job today is to talk about how we've used P integration at Proof. So again, we talk about this NXP structure where N is the samples and P is the predictors. In this case that I'm going to talk about, it's genes. So the integration across the predictors P is known as MINT, which stands for multivariate integration. And what MINT does is it performs simultaneous batch correction and classification, and then it integrates the data on the variables. So you can imagine that this might be useful for things like multi-center studies, where they haven't implemented a necessarily proper randomized block design, but you have similar data that's obtained at different times measuring a variable or predictor of interest. So it can be pretty useful for something like a meta-analysis, which is what I'll talk about in the upcoming example. So I'm currently working on a systematic review and meta-analysis of transcriptomics for idiopathic pulmonary fibrosis, which is a severe lung disease caused by excess fibrotic activity in the lung, which makes it difficult to breathe. So I reviewed 5,337 studies with a colleague, and we identified 11 studies that looked at gene expression. We then integrated these using MINT and then found a 17 gene signature of IPF. So this is how it looks like. Out of the 11 data sets that we identified, we split up 10 as our training set and left 1 out as a test set. Because this is P integration, we had to find variables that were shared between all the data sets. And so in this case, we identified 13,226 genes that were measured by each of the studies. So we've integrated the studies just based on those genes. We developed a classifier on the training set, and you can see that in the figure on the top right that this model kind of separates IPF from healthy lung samples pretty well, using only 17 genes on the first component. And on the bottom left, you can see that the AUC of the training model is 0.9491, which is really good. An AUC of 0.5 is basically a coin flip, and the closer you get to 1, the closer it is a perfect classifier. And so when we test this classifier on the left out data set, we can see that it has an area under receiving operating curve of 0.9494, which is really good. So we can rest easy knowing that these 17 genes are pretty robust in their IPF classification mechanism. So looking into the biology of these 17 genes, we find a lot of similarities with what's been reported in the literature, which makes sense. So I've highlighted in this pickle green color the upregulated genes, which are all related to extracellular matrix proteins. And I'll highlight this one gene in bold called MMP7. So that's matrix metalloproteinase 7, and it's been studied extensively in IPF as a biomarker. In fact, one of the very first gene microarrays ever published used IPF as a model and identified MMP7 as being upregulated. So we have some confidence that this model is actually pretty relevant and accurate. So something that's a bit more interesting is what's downregulated, which I've highlighted in yellow. So a lot of papers tend to focus on the upregulated genes, and they miss some of the biology from what's downregulated. And so when I inputted these 7 downregulated genes into a pathway analysis tool, I found pathways related to the nervous system, which is strange because it's a lung disease. But when I did some more digging, 4 of these genes are actually highly expressed in alveolar type 1 cells, or AT1 cells. And this one gene called MYRF, or myelin regulatory factor, is actually speculated to be a key transcription factor in AT1 cells. So from what we understand of the biology of IPF, the AT1 cells are injured during the fibrotic process. And so we're likely picking up a sort of AT1 damage or depletion signal with this classifier, which is pretty interesting. And so that's an overview of what P integration is. And at this moment, I'm going to stop my screen share and hand it over to Dr. Singh.
So it's great to see a good turnout today. It's about like 43 people on the line, and it's nice to see a lot of the trainees on the call. So the goal of the second half of the talk will be to look at the other side of the coin. And we have termed this N integration, which is essentially the exercise of collecting a lot of data, different sources from different sources on the same set of samples. So a lot of the material I will show you today will really be a primer on N integration and not really be details in terms of the algorithms and the mathematical formulas and so on. So it's really to get you interested in multi-omics data integration and determine whether or not it is applicable in your research. So I'm going to start off with this diagram that you see here of this hypothetical biological process that's occurring. And as you can see, there's a lot of different biological mechanisms focused on these different compartments in play. So I don't think I have to convince anyone here that biology is very complicated. So we have to sort of look at these various different modalities when we're sort of interested in disease mechanisms. And so the reason why we are interested in multi-omics data integration is really to get a holistic view of the biological processes that may be occurring. And you can see this depiction of these, you can think of these as spreadsheets in an Excel file where you have, when you're measuring these different variables along one axis, and then you have your samples on a different axis, and you can sort of do these for different modalities. And now I'll show you some examples that proof has been involved in and show you what we found. So I'll start small, and I'll start with a single molecule biomarker to begin with, and this is anti-proBNP, which is an excellent rule-out indicator for heart failure. So you can, as you can see from this meta-analysis plot on the left here, the sensitivity for anti-proBNP in ruling out heart failure is quite high. We have almost a perfect sensitivity value. However, the specificity is quite low. And so one of the questions that I had during my postdoc was whether or not we could improve the performance of the single molecule biomarkers by incorporating additional variables, and in this case, just looking at other plasma proteins. And you can see the results on the ROC curves on the right. And so you see that anti-proBNP in this, this cohort, which was recruited at St. Paul's Hospital, and a subsequent cohort was recruited at in one of the Alberta sites. And you can see here the performance of anti-proBNP is quite high in discriminating heart failure with controls. However, if you incorporate these about like 12 plasma proteins into this predictive model, we're able to have this incremental improvement in the performance of this model over anti-proBNP. And one of the reasons why this may be occurring is because we're capturing a lot of these different mechanisms that may be at play in heart failure. And you can see here anti-proBNP, which is depicted by its gene symbol, and PPB, is also positively correlated with these other plasma proteins that are indicative of these other biological dysfunctions. So going from the previous slide was on a single biomarker, and now so we're looking at transcriptomics. So again, we're still at a single omics case here, and we're sort of building this up to more and more omics data sets. So this study is an asthma example. So here, this graph is showing you lung function decline on the y-axis, and then on the x-axis is the time after allergen inhalation. And you can see here all the mild asthmatics that inhaled the allergen, they have this initial decline in lung function to obtain that airway hyperresponsiveness threshold of 20%. However, about 50% or so, which is also what is observed in the literature, have an initial decline and then a recovery after about 2 to 3 hours. The other 50% have this chronic late phase response. And so the goal of this study was to really use the blood transcriptomics, so these mRNA transcripts, and determine whether we could identify some predictive models that could discriminate these responders before the allergen challenge. And you can see the performance characteristics of these models in this graph here. The clinical model, which has the usual variables like age, sex, and so on, is quite poor in terms of determining who will elicit what response after challenge. However, some of these molecular panels of mRNA transcripts have a much better performance in discriminating early from dual responders. And it's really this again, going back to this complexity of biology, the fact that these panels can sort of capture these different parts of this mechanisms may explain why they are better and more sensitive at discriminating these response types. So you can see this improvement that we're already making with one single omics data set. And if you can imagine, when we add other biological compartments, such as the proteomics, the epigenome, where we have this regulatory interactions between biological domains, that we can sort of improve upon what we're finding when we're looking at single omics modalities. So continuing on the asthma example, we also looked at the metabolome. So not only looking at the mRNA transcripts, but also looking at the metabolites. And in this case, we're interested in how these different molecules changed in response to allergen challenge. So we compared before and after, about 2 or 3 hours after allergen challenge. And what we found was a lot of the significant molecules that were changing were related to the lipid metabolism pathway. So a lot of the genes are these should be italicized, but these are significantly changed from pre to post challenge, and they change differently between the responders. And you can see the metabolites that are also changed are these lysolipids, which are often cleaved and result in these mediators that cause asthmatic symptoms. And what we also found was these highly correlated networks, and you can see one here, particularly in the dual responders, so the cohort of subjects that elicits that chronic late phase response, are these highly correlated gene metabolite networks. And this is depicted using this node edge framework. So the nodes in the circles are the different mRNA transcripts that you see here. The metabolites are in the rectangles, and the link or the edges that you see here are either positively correlated in the red or negatively correlated in the blue, as depicted by this color key, which looks like the flag of France, which is not due to chance, but the author of this algorithm is French, although she has moved now to Australia, so I'm not sure if she's still retaining her French citizenship. But again, focusing more on this network that you see here, some of these molecules are already known to be implicated in allergic asthma, such as IL-5 receptor, which is the receptor on eosinophils, and arachidonic acid is also quite well established in asthmatic disease. And so the goal here is we're sort of identifying these networks that have biological plausibility. So there's these other unknown sort of networks that may also have some novel implication in asthma, but that remains for further study. So now looking at another scenario with multi-omics and non-omics data integration. So in this case, we looked at a cohort of heart failure patients, and we incorporated molecular domains. So gene expression again from the blood and protein expression from the blood. But we also looked at sensor data, and this was using Holter monitors. And the purpose here was, again, can we improve on the clinical risk prediction models, which are quite, they're known to perform quite poorly in predicting hospitalizations in heart failure based on the literature. So this was really a pilot study where we looked at these various modalities and then identified these models and determined whether or not we could improve on the clinical model. And so we had a cohort of heart failure patients. We followed them over time, and then we tried to predict we tried to predict 3-month hospitalizations. So some were hospitalized, others were not. And as you can see from this ROC curve again, that depicts the performance of these models that we're identifying. The clinical model that you see in the green, these are, again, they might look funny to some people just because of all these kinks, but really this is due to the fact that these are average ROC curves over various rounds of data splitting. And that's just to enable us to determine whether or not there's some overfitting going on. So these are sort of robust to overfitting. based on our limited sample that we had. And as you can see from this, this table here, the clinical model has an AUC of 0.76. But as you start incorporating some of these molecular data sets, and this column depicts the number of variables that are part of these models, you can see that we're sort of improving upon the performance of the clinical model. You can see that the proteins here have an AUC of 0.82. But what's also interesting is when you combine many of these molecular and non-molecular models into one, you have a significantly improvement on your AUC. And so this really goes to show you that these different molecular modalities may be capturing independent sources of variability with respect to heart failure hospitalizations. So again, ensembling of these different decision makers can often lead to better performance. Now I'll talk about an approach that I was very interested in developing. And this is because a lot of the current machine learning and sort of the literature for developing these multidimensional models that incorporate these different data sets or these decision makers are often derived independently within each of these different modalities. So I was really interested in developing an algorithm that not only allows me to determine predictive molecules across these different domains, but also explains some of the common sources of variability that may be occurring across these different biological domains here. And so the purpose, or what you actually get when you apply this sort of algorithm, are these small set of highly correlated molecules across these different domains, but they're also predictive of various, in this case, categorical outcomes. And here we applied Diablo to a cohort of neonates during the first week of life. So we collected blood from this cohort, mostly African and Australasian, and we had 30 babies here, and we collected blood at day 0 and all the way up to day 7. And we profiled, again, going at a more and more omics data sets, the cytokines, metabolites, proteins, and transcripts. And one thing that was really neat, again, this is a collaboration with our group and Bob Hancock's group at UBC, and a lot of other Australian centers, sorry, not Australian, African centers, that they also looked at analyzing these different modalities independently and compared the results with Diablo. And as you can see here, once you apply these, sorry, I'm missing my cursor, these predictive models independently to these different domains and look at the relationship between the variables that are selected, you have a lot of this, this independent variables. So a lot of these single molecules that you see that are selected across these different domains are not really correlated with each other. And that's sort of what the purpose of this exercise is really doing, right? They're interested in the best predictors, regardless of what's happening between these different domains, right? So they're sort of missing that extra source of information. And then if you apply Diablo to this data sets, you can sort of reduce the dimensionality of a lot of these thousands and thousands of variables and sort of visualize what the molecular changes look like. And this is depicted by this scatter plot using these. So this is sort of, you can think of it as analogous to a principal component, but it is supervised and it incorporates more than one data set, right? So here, these different points are the individual babies over the first week of life here. And you can see these coordinated changes during that first week, which was something new that we uncovered using the study. And then, so this is the application of Diablo to this cohort data sets. and you get a much more blocky structure. And this is depicting this is a heat map visual depicting the correlation, both positive and negative correlation between the variables that are selected, also known as features. And what's also interesting is that in the network analyses, we get these sub-networks of highly correlated molecules that are actually attributed to common gene ontologies. And in this case, a lot of these are coming from innate immune pathways, which makes sense because, again, this is the first week of life. They're still, their immunity is still developing. So a lot of this is also in Africa, where tuberculosis is prevalent, right? So this really goes to show that we're sort of we're able to uncover these common mechanisms that span these biological compartments using an integrative approach such as Diablo, whereas the gold standard, or what we had in the existing literature, is really unable to do that. So I'll be remiss if I don't talk about single cell multimodal omics. And it was named the method of the year in 2019. And I believe single cell RNA sequencing was a method of the year in 2014. So we've come a long way in such a short period of time, where we can actually derive these multimodal omics data sets at the resolution of a single cell. And you can see these various different technologies that allow you to do these quantifications. And there's a lot of types of analyses that can be done with single cell data. And my colleague, Casey Shannon, talked about some applications in his previous proof webinar, which I believe maybe will be hosted on YouTube. So if you haven't seen that, please feel free to check it out. So again, I want to share examples of projects that Proof or I have looked at as well. And this study is looking at single cell RNA sequencing data, and also looking at the spatial coordinates of cells as well. And so as you can see from this, this panel A here, what the study does was actually quantify the relative expressions of genes, about 113 genes in different cells. And this was done with the cells localized to their position on the tissue sample. So we're able to quantify the expression at the location of these single cells. However, these technologies are still quite limited in terms of quantifying thousands and thousands of genes, which is possible in single cell sequencing data. So the purpose of this analysis was to look at spatial transcriptomics, which in this case is indicated by a platform called SeqFish, and then try to integrate this with the single cell RNA sequencing data that is much better at determining cell type identity. This heat map shows you the common genes between these platforms. So these are the different genes that were identified based on the single cell RNA sequencing data that are indicative of these different cell types. And there's a quite good overlap in terms of correlation between the RNA-seq data and the SeqFish data. And then so this model that was developed using the RNA-seq data was applied to the spatial transcriptomics data in order to overlay the spatial coordinates here of these various cells with the actual identities. So we can actually get now, using this expression data, the actual identities of these various different cell types. And other studies have also shown that the pattern of scatter may also be indicative of various diseases. So this is quite a novel advancement in technology. And recent technologies are also able to look at 3D locations and expressions of these different molecules. So I showed a lot of promise with the multi-omics data, but I want to also touch on challenges that may arise with these sort of efforts. And I've sort of put these four big topics that we often think about when looking at trying to integrate multimodal data. And the first one here is really at the pre-processing steps. So there's a lot of different experimental protocols that are being applied. Each has its own normalization strategies. So there's a lot of effort coming from from the single cell RNA-seq literature in terms of benchmarking different methods, whether they be through generating these data sets on artificial samples or using simulations. So benchmarking will be very important in determining how these different algorithms perform on these different data sets that are experimentally generated. Another issue that we also see is the scale in the data. So some of the data sets, for example, genotypes are on a categorical scale. Protein expression is on a continuous scale. So you sort of need different data transformations when you're trying to integrate these different data sources. The number of variables, so here in the size, differs across different data modalities, and that can impact the total variation. So there's a lot of scaling strategies that are often used between these different data modalities to make them more similar. Removal of batch effects, which are often like well established that they occur in these data sets. A lack of overlap is also a big issue that we see because oftentimes when you have a set of samples, you may not be able to quantify well the various modalities. So there's often a different number of samples that end up occurring across your different modalities or a different number of features. So you sort of are limited to what's in common, or you can also impute missing values, but then you're sort of assuming that there is some sort of correlation structure that exists that you can leverage. And again, I'm sort of talking at a very high level overview of these different topics. So if I welcome anyone that is interested in more details to look at this white paper that explains a lot of this material in detail. The last challenge I want to talk about, and probably the most important one, is the interpretation. And really, this is going to what are we actually finding? What does it mean? Is it real? And so there's a lot of, I guess, thoughts behind what can potentially be interpreted from multi-omics data sets. And given that this is quite an emerging field, there's still a lot of different vocabularies that are being used. So this was sort of Daniel's attempt to sort of mention some of the standardized definitions before the talk. So we don't want to confuse our viewers here. Also, the interpretation depends on the type of analysis. So if you're interested in biomarkers and predicting different outcomes, then you would choose other methods than methods that are interested in cluster analyses that don't look at your sample labels. Simulations are also important because they help you determine what a given method is uncovering, and really allow you to determine the limitations of these different multi-omics algorithms. Again, I didn't really mention the names and the algorithms of these different case studies, but I'm happy to chat further if anyone is interested. Lastly, I want to touch upon using curated information. So there's a lot of a priori knowledge that is already out there. And so a lot of the molecular networks or these new biomarkers that may be identified can definitely be interpreted in the context of curated information. So if there's some networks that don't really have any known biology, you can sort of incorporate relationships with known molecules based on these databases and sort of enrich that interpretability there. So one comment on where this is all going and the future of multimodal data integration. So hopefully I've convinced you that um, multimodal data is becoming more common. And so as the technology improves, we're sort of getting much better at collecting data. So there's a lot of efforts as using sensor data, both in academia and in industry. There's obviously advancements in omics data. Incorporating exposure data is also a missing link that is often unobserved in these studies. Electronic health records, again, is a massive data source that can be leveraged. So we're sort of becoming very, there's a lot of efforts, I would say, that is going towards harmonizing the data into these common standards, common ontologies and frameworks, so that we could sort of analyze these data sets together. So going forward, I think we're probably see more and more of these multimodal data sets, and we're probably going to see them collected over time, as we see with a lot of the um, continuous monitoring of either sensor data, clinical data within the healthcare. So as the technology advances, costs get cheaper, we're probably more likely to see these types of data sets arise. Lastly, a point on looking at where we are, looking at electronic health records. So we're at a time where we can incorporate, and the methodology exists with the recent advancements in deep learning, is really to incorporate these different types of data. So in this case, we have this continuous numerical data sets, for example, systolic blood pressure that's measured over time. Narrative notes, so doctor's notes, can be quantified and translated into numeric values. Static variables as well. So these are very flexible models in terms of the types of data they can handle. They can handle missing values as well. And so we're at a time where we can use these various data sources to predict clinical outcomes. And so the remaining missing links are really the stuff that we talked about today. So a lot of the omics data, environmental data, that can be sort of incorporated into these continuously learning algorithms, and they can continuously be changing their predictive outcomes. So this is on the lines of the social dilemma, if you've seen that movie, where a lot of these big tech companies are sort of, they have done this job already by collecting a lot of your data, but now they're actually exercising these predictive models. And this can also be done for the good in our healthcare system. So lastly, some conclusions then. So I hopefully I've convinced you that modal data is here to stay, and we're able to obtain this at the single cell resolution as well as within the spatial context. We talked both about P integration when you have the same variables measured on different sets of patients or samples. And we also talked about N integration or multimodal data integration. And really the aim there was to learn these holistic patterns that span across these different biological compartments in order to understand the complexity that arises in heterogeneous diseases. I talked a lot about challenges, and again, if anyone wants to discuss specific algorithms and approaches, code and so on, please feel free to reach out to me. And lastly, Proof uses and develops methods for data integration. And hopefully I've shown you a lot of the studies I talked about where Proof center efforts. So with that, before I close, pitch for next month's seminar that will be led by Jiyoung Kim and Zhuja Hollander on how to successfully develop biomarkers for the clinic. And with that, I'll thank you all for spending your Friday afternoon or morning with us. And me and Daniel are open for questions. Thank you.